proceed.
Thank you. Assalamu alaikum, ladies and gentlemen. I would like to initially thank the organizing committee to give me the opportunity to share my research. Actually, it's a large research, and I am going to share a part of this. We have already working on more than 1,000 patients who have undergone different types of chemotherapy in breast cancer, and mainly we work on the genetics. Basically, we can classify it as a medical genetics. Now, today's I would like to share a part of this study. Impact of GST M1. There are many genes, and I am focusing on this only one gene here. GST on gene genotype on taxane-based chemotherapy outcomes in Bangladeshi triple-negative breast cancer patients. Initially, there are some introductory slides. Global prevalence of breast cancer, you know very well it. I have collected the data from GlobaCan and different types of cancer prevalence, and the highlighted portion is the breast cancer prevalence in Bangladesh. This is actually official data, but unofficially the frequency is very high, as you know. Incidence, mortality, 5-year survival. Now we would like to with the main point, treatment individualization. And now we are going to individualistic treatment, precision medicines. The A's are coming for AI and treatment individualization. And the ultimate goal of this study, or this type of study, is to genetic and pharmacogenetic studies to establish the selection of treatment that is most likely to provide more benefit and minimal toxicity. Treatment specification, actually, today's study is a part of a study, and in initial studies, we have recruited 330 patients. Then it has increased to more than 1,000, and treatment response was evaluated on the basis of RACIST criteria, which was published more than 2 decades ago. And toxicity evaluation was done with the base of CTCAE, common terminology criteria for the adverse events, where there are 2 types of toxicities, general toxicity and severe toxicity. There are 5 types of toxicity, grade 1, 2, 3, 4, and 5. Grade 5 is death, due to chemotherapy-induced death. And this is very rare now, but actually we can classify it into broadly 2 groups. Mild toxicity or general toxicity in group 1 and 2, and severe toxicity in group 3 or 4. In our triple-negative breast cancer, in our total study, we have got more than 100 triple-negative breast cancer patients who have undergone paclitaxel-based chemotherapy. And actually, we have tried to use the impact of this genetic variation on the treatment outcomes. Genetic polymorphism, you know very well. Any kinds of genetic sense in the single position is known as genetic polymorphism. There are different types of polymorphism, like SNP, insertion deletion, and there are other types of variation also, that is copy number variations, single nucleotide polymorphism. This is the part of methodology, how we go for genotyping. We are not going to elaborate it. And this is the PCR conditions. So we have gone a PCR of this gene. So how we have done it, it is elaborated in the slide, as it will be more boring. That's why I'm not going to elaborate it. This is the part of, initially we go for PCR, then we go for enzymatic digestion, specific to identify the specific mutation or any kinds of gene actually available. Now the demographic distributions of initial studies. Actually, we have 330 patients initially. These are the demographic distributions of the patients. But this is in this case, all are not triple-negative breast cancer. Only 100 was triple-negative. Now, within the 100 patients, this is the result of the taxane-based chemotherapy. The patient who carries this mutation or who carries this gene have within 100, we have 43 patients who have this type of gene or uh, genetic coded protein, and this is the enzyme. And 57 patients was devoid of this enzyme, and that is GSTM1. And we have found that it is significantly associated with the response, Taxane-Based response. The patients who carry these genes comparatively goes less response. And now different types of toxicities, mainly hematological toxicities was significant. And initially anemia, and the anemia was significantly associated with the patients who have not this gene. So actually the patient who carrying this gene is non-responsive and highly toxic to the chemotherapy. In case of neutropenia, so this chemotherapy-induced neutropenia was also significantly associated with the patients who have not carrying this gene. The patients who have this gene, they have got more toxicity. So and finally, leukopenia also was significantly associated with this. And we have also more kinds of toxicities, but that was not significant due to the number of patients. So if we got more patients, it will also be significant. That is gastrointestinal toxicity and thrombocytopenia. We have also gone for these studies. We have got significant association with other type of genes. But in this case, we have not got this association because of the less number of patients. So we have not mentioned here. So finally, GST-M1 gene can be used as a genetic marker for treatment, for the selection of treatment regimen for triple negative breast cancer. You know, triple negative breast cancer is most of the cases, it is in some cases, not most of the cases, in some cases the treatments become failed. Young patient face more toxicities. So in this case, we have found that GST-M1 can be a marker. The patients who have absent this gene, who does not carry this gene, go for more toxicity and less response. And that's all. We have some limitations also, patient limitations and different kinds of study limitations. But if you can cover more patients, more than 1,000 patients, so we can go for a very conclusive remarks, very conclusive findings, and we can go for clinical practice also. Thank you.